thanks for joining this conversation. I've heard a lot of interesting and exciting things about how you have used Cytely and also about your research. So I'm personally very excited to just walk through these things. Let's just quickly recap a quick before and after state. So when was it that you and your team started using Cytely?
This happened through actually interaction of my student, my master student in the group who interacted with Carl, one of your company employees. I think about 3 months ago, I was introduced to Cytely and to your team members.
And how many in your team are now using Cytely or at least have used it or have seen it?
So currently I have 2 lab members, one postdoc and one master student who are using Cytely and have seen it.
Before Cytely, if I understand it correctly, you were spending quite some time to find cell nuclei in which there were viruses to be found. So is that a correct kind of summary of how it went down? And could you give us a sense of the time that you spent doing that, let's say, per sample?
Yes, of course. So first of all, let me say that even though we're talking about 3 months' time here, the insight that we got into our project and how the experiment should be designed and actually redesigned, a completely transformable experience. And I'll explain why. So for the past, I would say almost 5 years, we worked with a very unique system where we figured out a way to label herpes virus DNA inside a cell nucleus. So when DNA is being ejected from a virus into cell nucleus leading to infection, we can now, thanks to this labeling system, we can separate the signal from viral DNA from cellular chromosomal DNA in the nucleus. And this is done through unique labeling of the viral genome. Now, to be able to do this measurement, we first need to transfect the cells with a plasmid that has specific labeling, and then we have to infect those transfected cells with herpes viruses. And the mutated herpes virus, its genome will be pairing with the transfected plasmid, and that what gives us signal. The purpose of this project is to track viral genomes in the cells as they come in, and also progression of viral genome replication within the nucleus. So as viral replication compartments are growing in size and numbers, we can follow this versus hours post-infection. And this should allow us to separate between cells where virus infection is progressing. In other words, it's so-called lytic replication of a virus, versus those cells where virus genome is delivered into the cell by the virus, but replication is silenced, so-called latent infection. So we wanted for the first time to be able to quantify this, because there is essentially very little work is done on quantitatively understanding the mechanism and mechanics of herpes virus latency and the switch lytic and latent infection. So once we were introduced to Cytely, so for what I said, it took 5 years to figure out the design of this. Lots of struggle was we were transfecting the cells, and the efficiency of transfection is rather low. Then we were infecting those transfected cells, and the efficiency was even lower. So we had very few cells that were both transfected and infected at the same time, and really had no understanding and appreciation of numbers. What numbers are we talking about? It was all done qualitatively, visually on a cell dish, basically, when we looked at the fluorescent microscope in the field. And I had students struggling to get any numbers that could be quantified in a meaningful way. So we started to work with Cytely, as my student in my lab was introduced to this through sort of a random, I guess, conversation with one of your team members in the lab. And we got to try this software, and I've seen it at work myself as well. I was present during one of the demos. And instantly we could scan. I mean, it would take many, many hours for a student to sit in front of the microscope to evaluate possibly a few hundreds of cells during many weeks, several weeks. And after this few hundred cell evaluation, we would get data for about 3 or 4 cells being both transfected and infected. With Cytely, we could instantly identify, of course, thousands of cells, and we could get exact numbers, percentage of cells that are transfected and infected. And we learned, to our great surprise, that in some of those cases, once we had maybe 80% of cells that were transfected, but then we would get only 10% of cells that were both transfected and infected. And once we could quantify this and see the difference in numbers, we could figure out that there are cellular defense mechanisms for the specific cell type that we're using that prevent both transfected cells that prevent post-transfected infection. We figured out what was issue with the design of the experiment, and thanks to this quantitative insight, now we started to use different type of cells that have reduced immune response. And now we can finally start dealing with different numbers of cells and get reasonable statistics, because this particular project depends on having actually significant numbers, so we can evaluate and understand the difference between lytic and latent replication.
So if I understand it correctly, there have been a number of, let's say, unique insights that Cytely has helped you achieve. You mentioned that you came in contact with Cytely about 3 months ago. And so I'm interested in what was the time between that we actually practically started working together and this insight came to be?
I think within of running the software for approximately 1 month, we came to this realization. And now we have redesigned the experiment using a completely different cell line. We're collecting data at a whole new level when it comes to numbers. And therefore, this project now is moving forward. Before, actually, we could not collect enough data to make any sense of what's going on in the system. So I would say within a month time, we could quickly get very clear insight exactly how many cells are we looking at, how many cells are transfected, how many cells are infected. We could do this separate transfection, infection, and then both get which cells are sort of double positive and get exact numbers based on evaluation of thousands of cells as opposed to evaluation of a few tens of cells.
And so if we think about this from more of a, let's say, long-term perspective, how do you think this will change and transform the way you work and, let's say, the how your lab works?
This is a specific project which, like as I mentioned, it's not just sort of one of the side projects in the group. It has become a very central project in the group. There was previously a graduate student who worked full-time on this for 4.5 years. Now there is a new graduate student to be who has also spent about 2 years working on this with a postdoc. Actually, 2 postdocs in the lab involved. So significant number of, so to speak, manpower or people were involved in this project over a long time. Herpes latency dynamics and mechanics is a very important and it's a major research area in herpes virology and in virology in general. But that, and also that herpes it's one of the most latent viruses that we know of. It infects 90% of world population and latent in a major part of world population, except it's a major health issue in those patients which are immune compromised. And that's when the virus instantly starts replicating, causing significant damage. And there's no clear understanding how to suppress this active replication and push virus back to latency in compromised patients. So the holy grail in herpes research is to develop drugs which either suppress latency long-term or eradicate latently infected cell pool. So this is, in other words, central, very central research in herpes virology. And this became now this field was, I've chosen this field already when I worked back in the US in my during my previous faculty position at the University of Illinois, where we started work on this problem. And this is becoming one of the major projects in my group now. However, we've put a lot of time investment and funding, and with very limited progress to great frustration, because we could not be very quantitative. The purpose of this was to be quantitative about this.
Right.
There's quantitative data in terms of evaluating latency and see which parameters and how latency is triggered. I'm sorry, reactivation from latency is triggered. And now we can suddenly analyze thousands of cells after just recording the images for our data and looking at this data through Cytely application, we can instantly evaluate the progress of the ongoing infection in the cells and the switch between lytic and latent replication. I mean, I think that's finally we feel like this project is actually working, and we can learn very valuable information that will shape many different variations of this project for different lab members in the future.
Wow, yeah, that sounds like a transformative change, just like what you mentioned in the beginning. And it also sounds like it's a question of a magnitudal change. It's not an iteration or so. Like this is a whole new level of data gathering and the quality of the data, as far as I understand.
Absolutely. It's not an improvement. It's not a matter of being more efficient or collecting the data faster. It's basically a matter of transforming the knowledge that we gain from this research.
Have you come across any other solution or, let's say, workflow that could give you the same results?
No, we haven't. We actually, we were trying to digitalize some analysis of the data more in regard to understanding the extent of infection, not accounting the cells or looking at number of cells and so on. But once we identify an infected cell, using writing small script software in ImageJ analysis, we could quantify the extent of the fluorescent signal and convert it to the number of genomes within each cell. This is what we have done, but in the past. But to collectively get instant evaluation over large number of cells, we haven't come across a similar solution before. Absolutely not.
So if we think about, let's say, practical alternatives, so what would be, let's say that Cytely would just disappear. I hope not, but let's just say that would happen. What would be, let's say, the practical approach, and what would you expect in terms of outcomes of the alternative, let's say?
I see it as being essential now for us to move forward. I hope it does not disappear. Cytely, we figured out a very important thing, right? Thanks to Cytely, we could change our whole protocol. And thanks to Cytely, we can now use different cell line, etc. So we could entirely redesign, basically, very important part of the experiment.
Yeah.
And with this important change, possibly we could be more efficient at collecting the data, because we got very important insight. So we solved a big problem with Cytely. But I would say if we wouldn't have that access to Cytely, first of all, we would be struggling, and we would be at a different place right now. But also, I would say we hope to be able to continue working with Cytely, because we're just in the beginning, really, of this project. So it would be very key to success of this work.
Yeah, okay, understood. And of course, we're not going to disappear, and we would love to keep working with you. So don't worry about that. And so if you think about the future for a second, what do you think the impact of working with us will be in the future for your lab, for your group?
I have different ideas. What my lab does, so our research area is virology, but there's a specific type of virology which is called physical virology, and with physical, it's a term that has been minted maybe 20 years ago now, but it's really still relatively new and young field in virology. And it is about understanding physical interactions that are central to viral replication. And to learn about physical interactions, of course, one has to be quantitative. So once we want to be quantitative, looking at biological data, we need to work with large numbers of viruses and cells. To have this, if you work with single particle experiments, single particle experiments. And by single particle, we've been working with single viruses in our analysis. We've been working with single cells in our analysis, and different type of measurements like trapping cells in microfluidic chambers, using atomic force microscopy to look at single viral particles on a single cell nucleus, for instance. I think using Cytely to instantly now transform this knowledge into systems that or numbers which can relate to a real biological system, which is large in size. So, in the future, I'm thinking we could use Cytely to also analyze our atomic force microscopy data, mechanics data. We have a different type of signal. It's not a fluorescent signal; it's a mechanical signal, mechanical stiffness signal, but also learning not just of individual cells. I mean, we've been with AFM learning about a few tens of cells, possibly. If we can now analysis and large scans for hundreds of thousands of cells, would once again transform our knowledge in the field of virus-host interactions. Understand that at a completely different level.
Yeah, it sounds like we're approaching a whole new way of doing this type of science. From what I hear,
I think it's critical to what I think, in the field of biophysics. Biophysics now offers great set of tools, single molecule, single particle experiments, which become available in biology. But now to really bridge the gap in terms of the scaling gap has been met. But also when it comes to statistical numbers in single particle experiments, they're essential to be able to come with, have very good statistical analysis and systems which are large enough to represent real biological system, to put this in context of cell biology and infection.
As an enthusiast in this space, it makes me very, very encouraged and excited to see someone who is so experienced as you talk about the future in such positive terms and be so excited about this. For me personally, it just gives me the sense of that what we're doing is really, really important. And I really appreciate you sharing this.
Absolutely. This, it's, I think it's just tremendous. I'm really impressed.
Okay, good to hear. And just a few quantitative questions, because we have been talking about quantitative science a bit. So from 1 to 10, how much do you want to go back to your old way of working?
It's a clear answer. We don't want to go back to the old way of working.
Yes. Okay, so 0, I assume. And from 1 to 10, how much do you want to keep using Cytely?
Absolutely a 10.
Okay. And what can we do to turn that into an 11?
I just think that it can be in the future broadened to, like as I was mentioning, there are different tools have been used in cell biology, but also in biophysics, and these tools become more and more quantitative. And physical tools we're using, of course, fluorescence microscopy is one of the major tools, but other tools like atomic force microscopy, for example. And I think Cytely can certainly be used in combination with those other tools, sort of providing the powerful processing of a large number of data from just not imaging, but also other type of data, such as mechanical data. As I believe cell mechanics is also a very central part of understanding biological processes.
And so kind of to piggyback on that, so imagine if every researcher would have access to Cytely and get results like yours. What do you think would be the impact on the world and on research in general and on your field?
To me, I think the most important change would be that it would allow better communication between biological scientists and physical scientists in solving important biological problems. This communication is still limited. We're still using a different set of tools when we come from the side of physics and physical chemistry and biophysics versus cell biology and molecular biology. I think Cytely could bridge this gap in a sense and allow researchers to speak the same language when it comes to experimental analysis and from the experiments. So they at the level and value and context that is valuable on both sides, biology and physics, and that will be certainly can solve many more problems in the future.
Yep, no, agreed. And so if we think about, if we see this, let's say as a recommendation to use Cytely, why do you think someone listening should take action right now?
I'm positive that there are a number of virology researchers out there who are attempting to learn sort of meaningful information from their super resolution, high resolution measurements, and are lacking, was lacking this knowledge of availability or capability to process, once again, to analyze their system at a larger scale, could be missing out at possibly better design of their experimental approach system.
So it really comes down to being able to see, maybe like step outside of the box that we're in, using the, let's say, better data to give us a vantage point to let us understand, okay, how should we even approach this problem? Is that a way to look at it?
Absolutely. I think, that just like what happened in our case, without having access to this large amount of information that we quickly received now, we would be struggling figuring this out. It could have, taken much longer time, or maybe we wouldn't be able to figure this out at all. So it's hard to tell.
Yeah.
But at least, it has made a big change.
Okay. Yeah, thanks for sharing. And is there anything in specific that you think that we can improve?
I think, my students have been mainly working with software. I've seen the software work once, no, actually twice, one demo and one time actually at the actual microscope.
Yeah.
But all the details, my students are actually they're using the software itself.
Yeah.
So it's hard for me to say, when it comes to things like interface and so on. I'm sure you have scientists who are working on this in the team. So I can't really, provide any recommendations or wishes there. Or maybe it's great already, possibly. I know that I haven't heard anyone complaining in my lab.
That's typically good news.
No one said, we wish this was done differently and so on. I only heard very extremely exciting and positive feedback. Like I mentioned, when it comes to processing different type of signals, maybe that feature is available, or if it's not available, it would be great to have that feature. So it could be applicable and be combined with different instrument systems, not just fluorescence microscopy, possibly. This was just, maybe that, those options are already implemented or being implemented.
Well, I'm sure we'll hear it from Blase otherwise if we're missing something.
No, I haven't heard any other wishes, so to speak, except that it's really great, and the students were, very, very pleased and excited about using it.
Super happy to hear that, and it's been great to work with your team as well.